I'm George Demopoulos, and now we will show you a procedure on how to infect the Aedes aegypti mosquito with dengue virus. And the first thing we'll need to do is to propagate the virus in an Aedes albopictus cell line.
My name is Jose Luis Ramirez, and I am a PhD student in the Demopoulos lab.
For this procedure, we'll need a BSL-2 facility with a tissue culture hood, a CO2 incubator set at 37 degrees Celsius. We need 50 ml plastic conical tubes. We need human serum. We need a water bath at 37 degrees Celsius, a centrifuge. We need 96 well plates and other standard molecular biology and biochemistry reagents and consumables. So this is a regular tissue culture hood in a BSL-2 facility. And the first step is to take out the cells that have grown to an 80% confluency in a 75 square centimeter flask. This is an MEM media with 10% heat inactivated fetal bovine serum, supplemented with 1% L-glutamine and non-essential amino acids. Okay, now we will add the dengue virus to the cells. We have taken out the virus stock from the -80 freezer, and we had thawed it on a 37-degree incubator. We add about 0.5